Okay, everybody, I guess it's 9:30, so time to do my thing. So Paul's out of town today, so he asked me to come in and talk to you guys. So my name's Tom. I'm also a professor in physics and biophysics. And so today, Paul asked me to talk to you guys about diffusion, because part of my work is about studying intercellular diffusion. So I thought I'd give you a introduction to how we can quantify diffusion and a few actual experimental examples of when it's useful to think about. So it turns out that one of the, and by the way, please feel free to interrupt me at any time. So like if you got any questions or you want to say anything, just stop me and let me know. All right, so it turns out that one of the first places where people thought about diffusion in a biophysical context was thinking about a cellular membrane. Okay, so this is an example of a model for the cell membrane. Has everybody seen something like this before? So this is what's called the fluid mosaic model. Okay, so what it's made out of is the membrane itself is a bilayer of these amphipathic phospholipids. So the way it works is that these long fatty acid chains are hydrophobic, and therefore they want to cluster together, whereas the phosphate heads are hydrophilic, and therefore they want to be exposed to the water. And so these things self-organize in such a way that the fatty acid chains come together in order to hide themselves from the water, and the phospholipid heads are exposed to the water. And so what you wind up getting is a bilayer of this thing that it wraps all the way around the cell. Okay, now in addition to the phospholipids that make up the membrane itself, you can also find embedded in the membrane a variety of other things. So for example, here these things which look like green vegetables or something are membrane proteins. Okay, so these proteins can be things like ion channels or some kind of transporter, or things which actually manage the import and export of phospholipids into the membrane itself. So these proteins that are embedded in the membrane can do a variety of things. Okay, so now once people had this model established, okay, they knew that the membrane was made out of phospholipids and you could find these other things embedded in it. And then people started thinking about how this is going to behave. So I've kind of already given away the answer by telling you that this model is called the fluid mosaic model. Okay, so it turns out that the way this thing behaves is it's not really like a solid, rigid structure. That this membrane, even though it's a self-organized structure, also behaves something like a fluid. That is, all these phospholipids are more or less free to move around inside the membrane, and these proteins can also drift around inside the inside the membrane. Okay, so everybody cool with that so far? All right, so now let me show you some of the evidence that that is indeed the case, that this membrane, in fact, can behave like a fluid, and that these things are free to move around inside the membrane. Okay, so this is a pretty cool experiment. So what these guys did was they took a human cell, and they expressed some of these membrane proteins in the human cell, and they expressed them in such a way that they were fused to a green fluorescent protein. So that means that when you look at the cell under a fluorescent microscope, you can see the membrane is essentially coated with fluorescent green molecules. And then they did a similar thing to a mouse cell. Okay, so again, they took these membrane proteins and then they labeled them again with some kind of fluorescent dye. So in this case, it says it's rhodamine. So what happens is that the mouse cell is fluorescent in red, and the human cell is fluorescent in green. All right, then they used a cool little trick. There's a virus. I actually forget what the name of this virus is, but you can infect these cells with this virus, and it will actually cause these two cells to fuse together into one massive human-mouse monster hybrid cell. Okay? All right, so then what you can study is if these proteins are in fact free to move around inside the membrane, then they should be able to mix together like this, and you'll wind up with something that looks yellow, essentially, right? It's a combination of red and green. Whereas if the proteins are not free to move around, then they're going to remain in these essentially well-defined domains within this single monster cell. Okay, does that make sense? All right, so what these guys did was they performed this experiment, and they actually performed the experiment at many different temperatures. And then they quantify the number of cells that they measure that show up in this mosaic state. Okay, and so this is what they see. So on the x-axis here is the temperature, and on the y-axis is the percent, essentially the fraction of cells that they observe going into this mosaic state. And so what you can see is that this behavior, so it starts off at very low temperatures, essentially all the cells remain stuck in this state. And then as you increase the temperature, you see that suddenly they start to become mosaics. So what you see here is that this behavior where these proteins are able to move around and mix in the membrane is temperature dependent, okay? And one of the things I'm going to try to show you today is that this temperature dependence is in fact the kind of thing that you would expect if these proteins are able to move around the membrane. Okay, so now can anybody guess what's happening here? So why you go from no mixing to mixing all of a sudden? Okay, so the reason you see something like this is, again, this membrane is essentially made out of like fatty acid chains. So you can think of something like a stick of butter, right? So if you take a stick of butter and you cool it down, it's essentially a solid. And then as you heat it up, it eventually melts and the molecules are able to float around. And so that's essentially what you're seeing here, is that the membrane's undergoing a phase change. So at very low temperatures, the membrane is essentially a rigid frozen solid. And then at some point it undergoes a phase change where it becomes fluid, and then things can bounce around inside it. Okay, make sense? All right, here's another example of an experiment which implies that things are able to move around inside the membrane. Okay, so again, so this is a procedure that's called FRAP. This stands for fluorescent recovery after photobleaching. So again, what you do is you take a big honking eukaryotic cell, so these things are relatively large. And again, you can express proteins in them which are either fused to some fluorescent tag, or you can do some sort of chemistry in vitro to label these proteins with some fluorescent tag. But in either case, the end result is that you wind up with a cell where all these proteins that are embedded in the membrane are fluorescently tagged. Now, I imagine Paul has told you guys about photobleaching, yes? So if you take any kind of fluorescent molecule, if you zap it with enough laser power in order to try to excite it, eventually you're going to kill it. It's not going to be able to fluoresce anymore. So in this particular experiment, you do that on purpose. So what you do is you target a specific area in the membrane, and then you blast it with super high power laser. And what that does is it bleaches all the molecules that are within this region in the membrane that you targeted with the laser. So this is what it looks like on a microscopic scale. Where you zapped it with the laser, you get a bunch of bleach molecules which are no longer fluorescent, whereas outside this bleached area, the molecules are still fluorescent. Okay, so you zap them with this laser, and then what you do is you simply watch, right? So that's why this is called a fluorescent recovery after photobleaching. So what you do is you photobleach it, and then you watch after the photobleaching how the fluorescence in this region recovers. Okay, so now if the membrane is fluid, you would expect these proteins to be able to mix around and start flowing back into this region that you bleached, and therefore the fluorescence over time will recover. Whereas if it's not fluid, then you're going to stay in a state like this where you've just got this bleached patch of the membrane. And so it turns out that when you do experiments like this is the kind of data that you see, although of course it's highly stylized. It's pretty. So what happens is if you watch the cell like this before you bleach it, you've got some high level of fluorescence in this region of the cell, and then you blast it with the laser, and what that does is it bleaches everything, so the fluorescence drops suddenly, and then you watch it over time to see how the fluorescence recovers after that bleaching. Okay, and the fact that this fluorescence does indeed recover implies that these proteins are able to float around. Okay, make sense? Okay, now one more experiment. So all these examples I've shown you so far are like bulk right? So what we're doing is looking at large collections or ensembles of molecules and how they move around inside the membrane. So for example, here, essentially all we did was look for giant patches that were either green or red or yellow, right? And in this experiment, we looked for patches that were either fluorescent or not fluorescent, and then how those two patches mix together. So those are, in general, large collections of many, many molecules that we've been studying in these previous examples. Okay, now this is an example. I think this was done back in the 1970s or so. And so what you're looking at here is, again, it's a electron micrograph of a section of a eukaryotic cell membrane. And what these guys did is they took individual phospholipids that were in this membrane and then did some fancy biochemistry to them in order to tag individual phospholipid molecules with gold particles. Okay, and the reason that they use gold particles is that when you look at gold particles under an electron microscope, they're really electrically dense and therefore very easy to see. Okay, so now what you're looking at here is these little tracks are individual gold particles that they were able to track with this microscope. Okay, so each of these tracks represents the path of a gold particle that's attached to a single phospholipid molecule. So this is really like a single molecule experiment. Okay, so what you can see is that individual phospholipid molecules do not remain stationary in the membrane, that they move around. Okay, they don't stay in just one place, they bounce around inside the membrane. So now if you look at one of these paths in particular, okay, it looks pretty random, right? So it looks like it's a kind of a messy track. It's what we would call random, all right? But now, as a biophysicist, right, so when you say something looks random, that implies that you know what random is, right? So what we're going to try to do for the rest of the lecture is to write down a model that describes what we would expect if the motion was random. And then we can take that model and compare it to our actual experiments to see if the motions of various kinds of particles are in fact random or if they're some other kind of motion. So in particular, if you track, in this case, a phospholipid, or in general any kind of biological molecule, you can ask, why is this particle moving? Is it moving as a result of random motion, or is it being actually actively trafficked through the membrane or through the cell? And in particular, you can ask, how is it moving? That is, you can analyze the details of this track, and by quantitatively modeling what you would expect this track to look like if it were random or based on some other process, then if you quantify how it's moving, that will, in principle, tell you something about why it's moving. That is, what are the mechanisms behind why this thing is bouncing around. Make sense? Okay, cool. So in general, what we're going to do is we're going to write down a theoretical model that describes what we would expect random movement in a fluid membrane or in general to look like. And in particular, it's going to be a mathematical model because we're biophysicists, so that's what we're going to do. Okay? Then we're going to perform experiments. So like I just showed you, we can track the motion of phospholipids in the membrane, or I'll show you another experiment where you can track RNA particles bouncing around inside cells. And then if we have time at the end, I'll show you one of my own experiments where we can watch transcription factor proteins bouncing around inside cells. So we can measure all these different kinds of molecules moving around in the cell, and then measure the parameters that are relevant to our theoretical model, and then compare what we get from our experiment to our theoretical model to see if it is random. Okay, so this is something called the scientific method, right? So you may be familiar with this. All right, so let's try to do this. Okay, so this is going to be the kind of model that we construct. Okay, so we want to construct a model which represents random motion of some particle in a fluid. Okay, so now when I say random motion in a fluid, what do I mean by random motion in a fluid? So it turns out that a good starting place as an example of random motion in a fluid is something called Brownian motion. Okay, So this was first observed by, as you can guess, some guy named Brown back in 1827. And he found that if you take some small molecules and you suspend them in a fluid and you watch them under the microscope, that they do not just sit there. So this is an example if you take milk. So this is just milk, and then you look at it at relatively high magnification under the microscope. This is what you see. So there's water, okay, that makes up the milk, and then there are particles of milk fat that are suspended in this water. And if you watch the milk fat under the microscope, you can see that these things don't just sit there. They jiggle around, right? Okay, and this jiggling, this motion of these particles is called, this is Brownian motion. The fact that when you look at small particles in a fluid, they don't just sit there, they bounce around. Okay, so now you see this kind of motion anytime you suspend any kind of small particle in a fluid. Okay, so now the question is, you see this random motion, what is it that's causing this random motion? All right, so why are these things moving? And does this behave? So is this kind of random motion, is it similar to the kind of motion that we see of these proteins or phospholipids in the fluid membrane that I showed you previously? Okay, so it turns out that the explanation for why Brownian motion exists was first explained by our old friend Einstein back in this, I believe this was his miracle year when he did all of his famous stuff. And another guy named Smoluchowski in a year afterwards, and then Langevin actually, these two guys, their explanation was pretty mathematically painful, whereas Langevin's a little bit simpler and easier to understand. But in general, what all these guys showed was that you can understand Brownian motion as a result of random collisions of these particles, whatever particles it is that you're looking at, that the motion of those particles is due to the collision of those particles with the molecules of the fluid that it's suspended in. So essentially what's happening is that you've got, whatever particle it is that you're interested in, and then there's the particles of the fluid, and then these particles of the fluid bounce off of it. And when they do that, they impart some impulse to this, or they change the momentum of this particle that you're looking at, which then causes it to move around inside the fluid. Okay, in particular, it's relatively easy to see that this explanation, first of all, explains the kind of temperature dependence that I showed you in the first slide or two. Okay, and the reason for that is that when you look at the velocity distribution of the particles of the fluid, people have known for an extremely long time what that should look like. The velocity distribution of these particles should just be a Boltzmann distribution, right? And in particular, their kinetic energy is proportional to their root mean squared velocity. So this just says that the energy of the fluid is equal to the kinetic energy of the molecules, okay? And in particular, we know from thermodynamics that the energy of a fluid is also proportional to its temperature. So in other words, you can rearrange this expression to show you that the root mean squared velocity of the particles in this fluid depends upon the temperature. That is, as you increase the temperature, you're going to increase the root mean squared velocity. Or if you look at the distribution of the speed of molecules, what you're going to see is something like this. So this is at a low temperature. You see most of the molecules have a relatively low speed. And what this VRMS is telling you is it's a kind of average of this distribution. Okay? So what you see is that as you increase the temperature, this RMS speed gets bigger and bigger. And in particular, the velocity distribution of the molecules spreads out towards higher and higher speeds. And as a result of the higher speeds of the fluid molecules, when they slam into your particles, that's going to change their momentum and their path more than it would if they were traveling more slowly. Okay, does that make sense? All right, cool. So, yeah, like I said, this is a rough explanation for why you see this kind of temperature dependence when you look at Brownian motion. So that explains this sort of temperature dependence up here. That doesn't explain this phase change. You got to do some relatively high-powered statistical mechanics to see the phase change happen. Okay, so everybody with me so far? Awesome. Okay, so now let me actually explicitly write down a mathematical model to describe what it is we would expect for some kind of random motion. Okay? So the kind of mathematical model we're going to have in our mind when we talk about random motion is something that's called a random walk. And in particular, the example I'm going to give you, just to keep it simple, is going to be a one-dimensional random walk. Now when you look at, for example, these phospholipids moving around in a fluid membrane, that would be two-dimensional motion. So you would just take the same argument and then extend that to motion in 2 dimensions. But it's still going to be fundamentally the same model. Okay, so a random walk looks like this. So here's me. I'm relatively skinnier in this picture. So now what I'm going to do is plot my position. Okay, so I'm going to stand here. So my position is going to be indicated on the horizontal axis. So I'm going to essentially just move back and forth like this. And on this axis, I'm going to have time. Okay, so I'm not going to actually physically move in this direction. That's just going to be the flow of time. So now what's going to happen is I'm going to start off here at t equals 0. I'm going to start off at a position that I call x equals 0. So this is just going to be my initial position. Okay. Now what I do is I wait for some amount of time, delta t. I just stand here. And then after an amount of time, delta t, take a coin and flip it. Okay. And if the coin comes up heads, I'm going to go in one direction, this way. And if it comes up tails, I'm going to go the other direction. All right. So now if you do something like flip a coin, then the probability of getting heads or the probability of getting tails is going to be 50%. I'm either going to get head, and I'm going to call the probability of getting heads, I'm going to call that P, which is equal to 0.5. And the probability of getting tails, I'm going to call that Q, which is also equal to 0.5. Now you can also imagine processes where these probabilities are not necessarily equal. Like you could have a weighted coin or something where this is like 90% and this is 10%. But just for the sake of simplicity, let's keep it as equal probabilities at this point. Okay, so I flip a coin, and the outcome of that random coin flip is going to determine which direction I go. So let's say I flip a coin and I get heads. So that means I'm going to move this way. Okay, and I move from my original position by a step size that I'm going to call L. Okay? All right, so now I've moved over here, step size L. The next thing I do is I wait another amount of time equal to the same delta t. So I wait again, and then after that same amount of time, I flip a coin again. So it's again a random process, and the outcome of that random process determines what I do. So let's say that in this case I got tails, so now I'm going to move back over here. Okay, and then a random walk is you just repeat this process over and over and over again. Okay, so now I wait another amount of time, delta t. I again undergo some random process that determines which direction I travel. So in this case, I go this way. Okay, then I do it again. You can see that I fell in love with my PowerPoint animations at this point. Okay, and then you do it again, and you wind up at some final position. Okay, make sense? So what you're doing is you're following a path, and the path that you follow is determined by the outcome of some random process. So in this case, the random process we're talking about is flipping a coin. In the case of Brownian motion, the random process I'm talking about is the collision of the particle I'm measuring with the particles of the fluid. Okay? All right, so now, how do I describe this path that my random walker took? Okay, so first of all, I know that if I took in steps, so in this case I took 4 steps, then the total amount of time that that random walk took was the number of steps that I took in times the time interval of each step. Okay, so this is going to come in handy later, so you should remember this. So this is the total amount of time the random walk took. I can also quantify my total displacement. Okay, so I'm going to call my displacement from my original position, I'm going to call that x. Okay, and I can express the total displacement that I underwent in terms of both the total number of steps that I took and the total number of steps that I took in only one direction. All right, so this m here is what I'm going to say is the number of steps that I took to the right. You can also do this by saying it's the number of steps to the left. You just pick one and stick with it. So I'm going to stick with the number of steps to the right. Okay, so I'll leave it as an exercise to the reader to show that this x is equal to quantity 2m minus n times L gives you your displacement. Okay, everybody cool? Unless you actually want me to explicitly. So I mean, this is really very simple, so I'll let you guys figure this out. All right, so now this is our model for a random walk. That is our expected kind of motion that we would get if the motion were random. So now the question is, how does this compare, or I mean, how does this relate to what it is that we originally wanted to talk about? That is Brownian motion. So the idea is that this delta t, the waiting time, is essentially the time that you would expect between molecular collisions of the particle that you're interested in and the particles of the fluid. So this is the average time between molecular collisions, and L is essentially the mean free path of your particle between those two collisions. Okay, so you'll get one collision, and after that collision it's going to travel some length L, and then after it travels that length L, it's going to encounter another collision. Okay, make sense? All right, cool. All right, so this is the model. Now let's see how we can make predictions using this model. Okay, so the first question I ask you is, if this is our model, for an individual walker, can we predict what this displacement is going to be as a function of time? What do you think? Well, so the answer is no, you can't. It's because the process that determines the path that you take is random. That is, in this case, it's a coin flip. In this case, it's molecular collisions. So because it's the process that determines the path is random, for each individual random walker, you cannot predict where it's going to wind up. Okay? It's not, in other words, it's not deterministic. Okay, so each individual random walker, you can't predict where they're going to go. So now I ask you, what happens if you, instead of looking at an individual random walker, you look at a group or a collection, or in physics we like to use the fancy term an ensemble of random walkers. All right, so what you see here is a little piece of MATLAB code that I wrote. So what you see here is this is a random walk. And I don't think you can see the fine details in this image, but it's a random walk that consists of 9 steps. Okay? So it's going to start over here, and it's going to end over here. And so what I wrote in this MATLAB simulation is it's going to perform this random walk, I believe it's 1,000 times. And so every time the random walk winds up in one of these little blue bins down here, I'm going to increase the height of that blue bin by 1 unit. So here we go. I'm very proud of this piece of MATLAB code. Okay, so you can see what's happening is that here towards around the same displacement where you started, you're more likely to wind up in the middle, essentially towards x equals 0, than you are to wind up out towards the extremes. Okay? Maybe I should have increased the speed of this a little bit. Okay, so you can see what's happening is it's building up this distribution of paths. Okay? So now if you let this thing run an infinite number of times, what you'll get is something that looks like this. So now this, what I'm showing you here, this is the distribution for the displacement. Now like I showed you on the previous slide, I can express the total displacement in terms of either the number of steps I've taken to the right or the number of steps I've taken to the left. So this is the same probability distribution except expressed in terms of the number of steps to the right I've taken. Okay, so what you can see is that the total number of steps I took, as I told you, was 9. Most of the time you're going to take half of those steps to the right, which makes sense because, you know, the probability of going either right or left is 50%. So on average, about half of your steps are going to be to the right, half of them are going to be to the left, and therefore on average you're going to wind up essentially going nowhere. Okay, but you can see that there is some spread to this distribution. All right, there are some walkers which which wind up out here. That's these guys. And there are some walkers which wind up way out here. So what this is a probability distribution, right? So this tells me essentially, even though we can't predict the individual paths of individual random walkers, if I look at an ensemble of random walkers, that ensemble of random walkers should follow this probability distribution. That is, if I look at some ensemble of random walkers, I'll find some that go very far to the left, some that go very far to the right. Most of them won't go anywhere at all. So what I can do is measure the statistics of the paths taken by many random walkers and see if they obey this probability distribution. And if they do, then that's what I will say. Then I will conclude, okay, those guys were moving randomly. Okay, make sense? All right, so now let's figure out what this probability distribution is. All right. So here are all the possible paths. If I take 3 steps, these are all the possible paths that you can take for 3 steps. Okay, so here, this path is I've taken 3 steps to the right. These 3 are I have taken 2 steps to the right and 1 step to the left. Okay, so in this case, I wind up 3 steps to the right. In all 3 of these cases, I wind up at over here, right? I wind up at this point. So all 3 of these cases wind up in the same place, but you can see that there are multiple ways to wind up here. So for example, I can take 2 right steps and then a left step, or I can take a right, then a left, then a right. Okay, so there are multiple ways to achieve the same outcome, right? Now, because each of those steps is random, okay, and I said the probability of going to the right is 50%, what I called P, and the probability of going to the left is 50%, what I called Q, then I can and I assume that those steps are independent of one another, then I can automatically write down the probability to get any one of these paths. Okay, so for example, here I took 3 steps to the right, and the probability of each step to the right is P. Therefore, the probability of this path is P times P times P, P cubed. Okay, the probability for this path is P times Q. That is P squared times Q. Okay? So in general, I can write down the probability for each path like this. It's the probability of going to the right raised to the power of the number of times I went to the right times the probability of going to the left raised to the number of times I went to the left. Okay? Very simple, right? Okay, now the next thing I have to do is take into account the fact that in general there are multiple ways to achieve each outcome. Okay, so for example, if I wind up taking 2 steps to the right, as I showed you, there are 3 ways to do that. And there are 3 ways to take 2 steps to the left, right here. Whereas there's only 1 way to take 3 steps to the right and 3 steps to the left. Does that make sense? Okay, so now to take that into account, I have to multiply by a factor which tells you how many different ways there are to combine these elements. Okay, and that factor looks like this. This is what's called the binomial coefficient. Okay, so again, n is the total number of steps that I've taken. So this is n factorial. Everybody knows what a factorial is, I assume. This is m. This is the number of steps to the right that I took. And so now if you figure this thing out, so for example, here I've got n equals 3. If I plug in n equals 3 and m equals 2, okay, so that corresponds to these guys. If I plug in n equals 3 and m equals 2, this coefficient will work out to be 3. That is, it's the number of ways that it's possible to take 2 steps to the right and 1 step to the left. Okay, so what this expression gives me is, this is the probability of taking m steps to the right. So it's the probability of taking some combination of m steps to the right times the number of ways to get that outcome. Make sense? Okay, this thing right here, if you plot this for different values of m, that is this, okay? This probability distribution that I showed you on the previous slide that we got from looking at a distribution of random walkers, it is this thing plotted for different, so these are the different values of m. You plug those different values of m into here and you get this, okay? Okay, now this function is what's called the binomial probability distribution function. Okay, and the reason why it's called the binomial probability distribution function is, so let me show you a few properties of this thing. So one thing is if you take so a binomial looks like this. This is a binomial. This is what in math you call a binomial. Now, if you take this thing and you expand it out, that is, so like for example, if I took p plus q squared, and I expand this thing out, what I would get is p squared plus 2pq plus q squared, right? And I can do a similar thing for p plus q cubed. And in general, if I do p plus q to the n, does anybody know what you get? You get that. Okay, what you get is this. Excuse me. And you sum over all m. Okay, so when you expand a binomial, this is what you get. You get this thing. And in particular, in this case, because we're using p and q to represent probabilities, and p and q are both equal to 50%, then this thing, when you sum up all possible values of m, it's equal to 1. Okay, so this is what we call, this distribution is normalized. Okay, and this is going to come in handy in a couple slides. Okay, everybody with me so far? Awesome. All right, so now, in physics or statistical mechanics, once you know what the probability distribution is that describes your process, you already know everything about that process. It's just a matter now of extracting information out of this probability distribution. So now that we know what this distribution is that describes a random walk, we're done. We can predict everything about a random walk. So now let me show you how you get information out of this probability distribution. And in general, the way that you get information out of a probability distribution is by taking averages. That is, finding the moments of the distribution. Okay, in particular, we can find the first moment. That is the average displacement. So if we calculate this will tell us how far on average does a random walker go. Okay? Now, to make sense out of this calculation, so first of all, remember that I showed you that we can express the total displacement in terms of the total number of steps and the total number of steps to the right. Okay? Now, the reason why this is useful is because this probability distribution that I gave you here is in terms of number of steps to the right? It's not in terms of total displacement. So I'm going to use this, essentially. So when I calculate the average displacement, I'm going to calculate the average of this whole thing. And in general, n and L are going to be constants. So the only thing I have to calculate the average of is m. And I can do that explicitly using this probability distribution. Okay, so let me show you how. Okay, so to show you how, we need to, again, know a few tricks. So where should I start? So first of all, everybody knows how to take an average, yes? So for example, if you guys took an exam, the way that you could calculate the average exam score in the class would be that I would take everybody's score. So like, let's say you got a score S1 and you got a score S2, so on and so forth, right? And then I divide that by the total number of people taking the exam. So this would be the average score for the class, okay? Now another way to write this is, let's assume that several people got the same score. So let's say that a number N1 got the score S1, okay? And then a number N2 got the score S2, so on and so forth. Okay, so now all I'm doing is taking individual people's scores, and if they're the same score, I add them together. Okay? and I still divide by n. Okay, now another way to write this is like that, right? And now this right here, it's the number of people who got score S1 out of the total number of people. This is the probability to get S1. And this is the probability to get S2. So on and so forth. Okay? So in general, to calculate the average of the score, what I would do is this. Right? This is just a shorthand way of writing this. And it turns out that in general, to calculate any average, this is what you do. If you know the probability distribution that describes that variable, you take the thing that you want to find the average of, you multiply it by that probability distribution, and then sum over all possible values. So for example, if I wanted to find the second moment, s squared, I would do exactly the same thing. I would multiply the probability distribution by this thing I want to find the average of, and then sum over all possibilities. Okay? All right, so this is one trick we need to know. The other trick, so let me show you what it looks like before I show you the other trick. Okay, so that's what I want to do. To find the mean displacement, I need to find the mean number of steps to the right from my probability distribution. And I can find that by using this general way to calculate the average of something. So the mean value of m is just m times p of m summed over all possible m. Okay, so the next thing I do is I substitute in p. I know what p of m is. It's this thing. This is p of m, so I write that out. Okay, now if you look at this, okay, that looks an awful lot like this, right? And we know what the answer to this is. That's 1. Okay, but it doesn't quite look like this because there's this extra factor of m. So we got to be clever to get rid of this extra factor of m. Okay, so let's be clever. So let me ask you this. I've got p to the m. What do I get if I take the derivative of that? Okay, I know that nobody's actually going to respond to me. So I'll just tell you what it is. It's m p to the m minus 1. Okay, ooh, that looks tantalizingly close to this, right? Except we're missing a factor of p. So I just multiply by a factor of p. So in other words, m p to the m is the same thing as p ddp p to the m. Okay? So in other words, if I take, how do I want to say this? So I'll say it just by pointing to the answer. Okay, so in other words, I can express this m p to the m just exactly like this. I can replace this factor of m essentially with a derivative with respect to p. So now I just take my binomial probability distribution and I rewrite it as a derivative with respect to p, and that gets rid of this extra factor of m. Everybody see that? Okay. Now, this thing, I know the answer to that. The sum over the probability, binomial probability distribution looks like this. It is a binomial, right? So this sum gives me p plus q to the n. Okay? I think I forgot to take a derivative somewhere. Okay, so this sum gives me p plus q to the n, and then I take a derivative with respect to p, which I think I forgot to write in here. You'll have to forgive me. But the answer you get is still this, okay? If I did remember to put in that derivative, and you worked everything out, and you remember the fact that p plus q is equal to 1, you get it's this, n times p, which I think makes sense, right? So this, again, this is the average number of steps to the right that you take, right? And what that is the total number of steps times the probability to take a step to the right. Makes sense, right? So in other words, if you take 10 steps to the right, or excuse me, if you take 10 total steps, this equation tells you would expect 5 of those on average to be to the right, which I think makes sense, yeah? Okay, so this is the mean displacement. Now if you take this answer for the mean value of m and you plug it back in here to tell you what the mean displacement is, this is what you get. Okay, so this is this, and then I plug this result into here using the fact that I know that p is equal to 1/2. Okay, so p is equal to 1/2, so I get 2n over 2 minus n gives me 0. So on average, a random walker goes nowhere. Make sense? That's because half the time, half the random walkers are going to go to the right, half the random walkers are going to go to the left. On average, they're going to go nowhere because it's random. Okay? All right, then the next thing we can do is calculate the second moment. And the reason why we want to calculate the second moment is okay, so again we had this probability distribution. This is the number of steps to the right, and this is the probability distribution, and we saw something that looks like this, right? So what this tells us, or what the average of m tells us, is essentially, it's kind of a measure of where the center of this distribution is. By calculating the second moment, what the second moment tells you is essentially how wide the distribution is. Okay, so if you guys are familiar with the standard deviation, the standard deviation is related to the second moment. Now let's see if I can do this without embarrassing myself. I believe it's this. Okay, so if you know the average, the mean, which we just found out, and you calculate the second moment, then that'll tell you something about the standard deviation. That is how wide the distribution is. Okay, and then, for example, the next thing you can do is calculate the third moment, and that'll tell you, again, something about how it's shaped. So once you know the probability distribution, you can extract information about that distribution by calculating the moments. All right, so let's calculate what the second moment is. And again, that'll tell us something about the spread of the distribution. All right, so I'm going to follow the same trick. If I want to calculate the average displacement squared, I'm going to express that in terms of the number of steps to the right. So I take essentially this thing that I wrote down before, and I square it, and then I take the average, and I get something that looks like this. Okay? So to calculate the mean second moment of the displacement, I need to know the average number of steps to the right and the average number of steps to the right squared. So now let me show you how to get this. Okay, so it's exactly the same trick, except instead of calculating the sum of m p of m, it's going to be m squared p of m. Okay, and then to get rid of, so now you got to get rid of 2 factors of m, and so you just repeat this trick twice. So you're going to wind up taking 2 derivatives instead of 1. But it's the same trick, just performed twice. And when you do that, this is what you want. I'm going to spare you the horrible details. And when you do that, this is what you get. Okay? You get that the average displacement squared is equal to the number of steps that you took times L squared. Okay, now you'll remember that I showed you that, where should I write this? I'll write this over here. So now usually when you perform, whoa, that's cool. So now usually when you perform an actual honest-to-God experiment and you measure the motion of some particle through space, you're not measuring the number of steps that it's taking, because simply you can't see the number of steps that it's taking. What you're doing instead is watching the random walker over time. So instead of expressing this answer in terms of the number of steps, let me express it instead in terms of the total amount of time that it took. Okay? So now let's see, can I do this? So I showed you before that the total amount of time that a random walk takes is equal to the number of steps times the time interval of each step. Okay? Now, how else? So then if you take this result and you plug it in here, you substitute it in for n, okay, and then you'll wind up getting something that looks like this. 2 times L squared over 2 delta T times T. So this T here, that is this, okay? Delta T is the time interval between steps, and L squared is the average size of each step. Now, this combination, so this is the size of the average size of each step divided by 2 times the time interval between steps. Both of these things are constants, right? So I'm just going to take this collection of constants and group them together and call this a new constant. In particular, I'm going to call this group of constants something called D. Okay? And this D is what is called the diffusion constant. Okay? So whenever you hear people talk about the diffusion constant of some particle through a fluid, this is what they're measuring. Okay? Yes, please.
Why did we add the two? It's like I get how we did it and how it works, but like, what's useful about that? Why did we do that?
Okay, the reason why we did that is, so this is a random walk in 1 dimension. And it turns out that if you repeat this process for a random walk in 2 dimensions, what you're going to get is something that looks like this again, except multiplied by a factor of 4. So essentially what I'm doing is I'm defining this diffusion constant in such a way that this definition remains consistent when I perform the same analysis for different numbers of dimensions. So I'll show you what the general result is for different numbers of dimensions in I think it's on the next slide actually. Okay, so this is simply to make things, and another way to think about this is you can also derive this result in terms of a differential equation called the diffusion constant. And when you write down this differential equation that describes the same process, you wind up getting something that looks like this constant. And again, to make the definition of the diffusion constant with that differential equation agree with this definition of the diffusion constant, this is how we define this thing, okay? Okay, now, so this is the result that we get. This is the important thing right here. That the average of the squared displacement is equal to 2 times the diffusion constant times time. So in other words, the longer you watch it, the bigger this number is going to get. Okay, so this is what you should remember right here. Okay, so this is relatively important. So now is everybody cool? All right, so essentially the rest of the lecture, we're just going to play around with this, all right? And I think I actually need to go to another PowerPoint presentation at this point. Okay, so just to summarize, what I showed you is that you can think of diffusion or Brownian motion in terms of a random walk that's caused by collisions of the particle that you're looking at with the molecules that make up the fluid. Okay, and this was the result that we got. That the average of the squared displacement is equal to 2 times the diffusion constant times the amount of time that you watch it. Now in general, so this 2 times d times t is the result for 1D random walk. If you repeated the same analysis in 2 dimensions, you would get that it's 4 dt. If you did it in 3 dimensions, you would get 6 dt, and so on and so forth. So this little number here, d, is the number of dimensions that you look at. Okay, so in 1D it's 2 dt, for 2D it's 4 dt, so on and so forth. And again, this definition of d that I made was in order to make this work out and also be consistent with another differential equation that I don't have time, or I can't imagine that you're terribly interested in it either. But it's to make that definition consistent. Okay, now this thing is cool. This equation is cool because you can use this equation to determine, when you do experiments, you can use it to determine whether or not motion is random, whether or not it obeys this equation. And you can also get this to give you rough estimates of expected transit times if you know that what's going on is diffusion. So for example, if you're reading some paper and they tell you we measured the diffusion coefficient of this protein to be blah. Okay, so for example, we measure a protein diffusion constant to be this value, 0.4 micrometers squared per second. Now what does this mean? What does this tell you about this thing's motion? So this, you can use this number combined with this equation to give you some idea of how this thing moves around inside the cell, okay? So for example, you can ask, if I know this is the diffusion constant, how long on average will it take this protein with this diffusion constant to travel 2 micrometers? So this is like the length of an E. coli cell, okay? Okay, so what you would do would be to use this equation, okay? So where's my piece of chalk? I know that average of x squared is equal to, so in one dimension it's just 2 dt. So the amount of time it will take to travel 2 micrometers is just x squared over 2d, okay? So I would plug in 2 micrometers here, squared, divided by 2 times 0.4 micrometers squared per second, okay? And you see that your units jive, yes? And what you wind up getting is 5 seconds. Okay? So you can use values of the diffusion coefficient to give you some sense of how long on average it takes things to travel inside a cell due to diffusion. So here, for this particular protein to travel 2 micrometers, it would take on average about 5 seconds. Okay? Does that make sense? Yeah?
What do you use if you're, or what do you use if you're protein diffusion in 3D?
So I mean, it depends upon the data that you have and the question that you're interested in. So I would say, so for example, something like a nerve cell. So a nerve cell, it's got like dendrites and then a long axon, and then it's got a synapse down here. Okay, you can ask a question like, how long does it take to travel down this synapse? So this synapse is approximately 1 dimension. I mean, you could worry about the motion in the y dimension, but it's not going to be terribly informative. So it depends upon, you know, the question that you're asking and the particular cell that you're interested in. So this example here would be something like this, where you're just interested in how long it takes to travel a 1-dimensional distance. Okay? Anything else? All right, what time we got? All right, I got about 20 minutes, I think. Okay, so now let me show you how you can use this result to analyze the motion of different particles to tell you something about what it is that's causing the motion of those particles. So this is the result that we got if the motion of your particle is diffusive. It should obey this equation. If motion of a particle is not diffusive, if it's ballistic, that is, if it's like a ball and I take that ball and I throw it with some velocity v, this is the equation it would obey. So this is essentially just the definition of velocity, right? So velocity is distance over time. So this is diffusive motion, this is deterministic ballistic motion. And so now let's compare these two results to see how they compare to each other. Okay, so in order to show them on the same kind of graph, what I'm going to do is take this equation and square it. So it looks like this, okay? Because over here I've got distance squared. I want to also have this look like distance squared. So now if you compare results, what you see here is that the average displacement squared should be proportional to time. Whereas here, if it's deterministic ballistic motion, the average distance squared should be proportional to time squared. That is, if you measure the displacement of an ensemble of random walkers, and then you calculate what the average displacement squared for them is, if it's diffusive, it will scale linearly with t. If it's ballistic, it'll scale quadratically with t. Okay, so you can analyze and quantify the motion of your particles and compare it to these equations to determine what it is that's causing that particle's motion. In particular, if I plot x squared versus t, if the motion is diffusive, this should be a straight line, right? So if this plot is linear, then my motion is diffusive. If this plot is a parabola, then it's deterministic and ballistic. Okay? Make sense? So let's go back to the example that we started with. So now if we perform that analysis for these gold particles that we looked at, So again, what we're going to do is at t equals 0, each of these particles is going to be at some initial position. And then we watch them for some length of time t. And after some length of time t, they will have moved to a new position. So that displacement, the distance between their initial and final displacements, their initial and final distances, that is the displacement. So we just measure that number for each of these particles, and then we square it, and then we take the average, and then we plot it. Okay, so we do that for each of these particles, and this is what you get. Okay, so this is x squared, mean squared. So this is, you'll hear this referred to frequently as the mean squared displacement, because that's what it is. And you'll also see people refer to it as the MSD, because that stands for mean squared displacement. So if you plot the mean squared displacement versus time, so this solid line here is what you would expect for for diffusive motion. It just has a linear response. And then each of these tracks are different experiments where they quantify this motion. Yes?
So ballistic, does it mean that it mean that it prefers to move in one direction?
So ballistic means that it will, if it travels in one direction, it will just travel straight. So this is ballistic motion. Okay? Yeah. So ballistic motion just means it's traveling in one direction with velocity v. Okay? All right, so you compare the data to what we would expect from diffusive motion, and we conclude that these are within experimental error. This is diffusive motion. That is that there's nothing deterministic. There's like no protein involved here that's dragging these particles along. It's just purely random motion due to molecular collisions. Okay? All right. So let me show you. Yes, please.
So to understand, is there any machinery working over there or not?
That's right.
You do this analysis and it's like, okay, since it's diffusive.
That's right. There's no extra machinery.
Doing by itself.
Exactly. That's right. So this, because it's diffusive, then you know, like for example, you know these things aren't attached to some other protein in the cell, right? Because if they were, then they'd be anchored there and they wouldn't be able to move at all. Or you could also imagine maybe these things are attached to like one of these molecular motors that Paul studies, and then you would expect to see something closer to ballistic motion. So the fact that this motion is diffusive means that essentially there's no other stuff involved. It's just these things floating around. Okay? Yeah?
So when you're calculating this, do you use two-dimensional?
So for this you would use two-dimensional, yes. And essentially all that would do, so again the equation in general looks like this. Okay, so when you plot this, this is the equation of a line. And this combination of constants just gives you the slope. So whether it's two-dimensional, four-dimensional, all that's going to do is just change what the slope looks like, but it's not going to change whether it looks like a line or not. Okay? Yeah?
So you use like the one dimension for like nerve cells, and like the 2-dimension for like other kinds of cells. Is there any time when we need to use the 3-dimensional? Is there any like
So,
that we do that?
so there are certainly instances where it would be useful, right? So if we have time, I don't know if we're going to or not, I'm going to show you some data that I took where you have an E. coli cell. Okay, and the E. coli cell is like a cylinder, and I tagged a protein inside the cell with a fluorescent protein, and then you can watch this thing bouncing around inside the cell. So now, depending upon how you actually do the microscopy, if you just look at it under the microscope, essentially all you have is information about how it moves in the XY plane. And in that case, you would analyze it using two-dimensional motion. Now, you can use fancier microscope that allows you to also determine its motion in and out of the focal plane. And so if you have information about that motion in that dimension as well, then you would think about it in terms of the 3D motion. Okay? All right. All right, so everybody's cool? All right, now let me show you one other way to look at this exact same equation. Essentially another way to show you what the difference between diffusive and ballistic motion is, and to show you that you can actually get combinations of these. That sometimes you can measure motion which isn't purely diffusive or purely ballistic, that it's some combination. Okay, so again, let's start with our equation for diffusion, which says that the mean squared displacement is proportional to time. And for deterministic ballistic motion, the mean squared displacement is proportional to time squared. Another way to think about this is, let me take the logarithm of both sides. Okay, so I take the logarithm of x squared, mean x squared, and I take the logarithm of t. I do the same thing over here. Now, if you remember your properties of logarithms, when I take the logarithm of something raised to some exponent, I can pull that exponent out front. So I get this. So over here for ballistic motion, the logarithm of x squared is proportional to 2 times the logarithm of time. For diffusive motion, the logarithm of x squared is proportional to 1 times the logarithm of time. Now, why do I care about this? Let me explain this in a second. Okay, so now what I can do, so again, this is the equation of a line, right? So what I have is the log of x squared is proportional to some constant, okay? And that constant depends upon how it behaves with respect to time times the logarithm of time. This, so if I plot this, I can plot this as my y variable. I can plot this as my x variable. And this is the slope. So what this exponent is will determine what the slope on a plot of log of mean squared displacement versus log time is. Okay, and why I care about that is, now if I have something which looks diffusive, or something which is diffusive, and I plot it on log-log axes, the slope of that should be equal to 1, because that's just telling me what this exponent on the time is. Ballistic motion will have a slope of 2, okay? So this is diffusive, this is ballistic, okay? So this is another way to determine whether your data are ballistic or diffusive, is you plot them on log-log plots. All right, everybody cool with this? All right, now, so this immediately raises another question. So here I have a slope of 2, here I have a slope of 1. Can I get things which are not, which have like between 1 and 2, or less than 1? And the answer is yes, okay? So in general, what you'll measure is that the mean squared displacement is proportional to some power of time. If that power of time is 1, then it's diffusive. If that power of time is 2, then it's ballistic or deterministic. If you get something between 1 and 2, that's what's called super diffusion. And so what that is a combination of deterministic and diffusive motion. So an example of this are the molecular motors that Paul studied. I don't know if he's talked about these in class. So when you measure the motion of these molecular motors, what you'll see is that most of the time they just travel in one direction. Sometimes they'll stop and stutter, and it's that stuttering that is coming from the diffusive component. Okay, but most of the time they travel ballistically. Okay, so this is super diffusion. Below, for m less than 1, that's what's called subdiffusion. And in general, subdiffusion is what you get when you have, it's just regular diffusion. That is, there's no other mechanisms involved in making the particle move. The only thing that distinguishes subdiffusion from diffusion is that it's being performed in a crowded environment. That is, there's like, you can think of it as like a diffusion inside a maze. Okay, so rather than just having an open room, you're diffusing around in some crowded open room, like there's a whole bunch of other people in there, or you're diffusing around inside a maze. Does that make sense? Okay, cool. So these are the different kinds of motion you can get. Deterministic or ballistic, super diffusion, so this is like molecular motors. Diffusion, this is purely random. And super diffusion, this is random motion in a crowded environment. Yes?
So on the previous data you showed, like, you said, okay, this is a diffusive.
Yeah.
You say like, okay, with except
Yeah.
some experimental error, we can say that, but I mean, like,
Yeah.
it wasn't quite linear,
Yeah.
it was low. So can you say it's subdiffusion?
So if
Like t to the power 0.9, for instance, instead of 1.
So if you perform that experiment enough times to get your error bars down to where they don't overlap with that line, then I would say yes, that deviation from linear behavior would indicate that that is actually subdiffusive. I don't actually recall whether they actually did that in that paper or not. I would have to go back and look at it. But now let me give you an example of something which is definitely subdiffusive. Okay, but to do this, how much time we have? Got about 10 minutes. Okay, so now the rest of the experiments I'm going to describe for you are experiments studying the diffusion of particles inside E. coli, a bacterium. Before I can describe those experiments to you, I need to describe to you how E. coli works. Okay? So this is what E. coli looks like under the microscope. They just look like cylinders, spherocylinders specifically. These guys are about a micron wide, and depending on how old they are, so this is like after they've just divided, they're about, I would say, 3 or 4 microns long, and then they can grow to get about 8 or 10 microns long. So they're pretty small. Now inside, so this is just, all you're looking at, this is like looking at me, right? This is the outline of my body, right? Now there's other stuff inside my guts, right? And we can see the stuff in their guts by using various kinds of cool fluorescent microscopy. So this is an example of where I stain the cells with a dye that binds specifically to DNA and then fluoresces. And so what you see here, these bright spots correspond to the DNA or the genetic material that's inside these cells. Okay? Make sense? So now in particular, let's say you look at this cell over here. You can see that it has what looks like 2 relatively well localized blobs of DNA. And what this corresponds to is the cell's got 2 copies of its chromosome. And the reason for that is it's going to divide, right? And when it divides, it's going to keep 1 copy for itself, and the other copy goes to its daughter. So this is 2 copies of the cell's chromosome. And if you take a closer look at this molecule, you'll see that the E. coli's chromosome is a big circular piece of DNA that's about 4.6 million base pairs long. Now, on that 4.6 million base pairs are a variety of different features, which I don't know how familiar you guys are with these features, so let me be completely explicit about what they are. So you'll find some sequences of base pairs called genes, okay, and then these genes are all the information that you need to encode a single protein. So each of these big blocks has the genetic information to encode a single protein. These arrows, this little thing here, this little green box here is what's called an operator. This is a specific sequence of base pairs, okay, just like a gene. But rather than encoding a protein, all this specific sequence of base pairs does is it's a target for binding other proteins. So some proteins will come in here and bind to this specific sequence, and that will control expression of these genes. And I'll give you an explicit example of this if we have time, which we almost certainly won't. Okay, this arrow here is what's called a promoter. And this promoter is a binding site for RNA polymerase. So what happens is the RNA polymerase comes in here and binds at this region, and then it can read these genes into RNA, and then those RNAs are translated into proteins. If there are proteins bound at the operator, so you can imagine a protein bound at this operator, that will prevent the RNA polymerase from getting here and binding to the promoter, and therefore by having other proteins bound at these operators, you can regulate whether or not the genes are expressed. Okay, so you should think of the genome as essentially like a library. It's just a big collection of books that give you instructions on how to build things. And each gene is an instruction on how to build one specific thing. So for example, a birdhouse. Okay, so now these instructions are very important. You don't want just any grubby person coming in off the street and messing up your book. So if you go to like some fancy library and they've got really important books, they in general don't let you take those books with you. What they insist is that you make copies. Okay, and the cell does exactly this. It takes the information that's encoded in this gene, and then rather than using the gene itself, it makes a copy. And the copy machine is a RNA, my animations got all screwed up. So the RNA polymerase binds at the promoter, it reads the genes and generates a copy, and this copy is in a molecule called messenger RNA. Okay? Okay, then because you're a scientist and you don't actually know how to build things, what you do is you take these instructions and you give it to somebody who does know how to build things. And in the cell, this is something called a ribosome. And again, you'll have to forgive me, my animations got all jacked up. But the ribosome will read this piece of RNA, and it will translate the information encoded in this RNA into a protein. And the protein is the equivalent of the completed birdhouse, okay? So this is in general how genes get expressed in E. coli. And in this particular case where this gene is actively producing protein, you say that that gene is in its on state. That is, it's actually producing something. And let me skip this, but this is an actual picture of, so each of these strands here is, so this strand here is DNA. Each of these strands is a piece of RNA. And so at the intersection between this and this, right here is an RNA polymerase. So this RNA polymerase is reading this DNA molecule and generating a long strand of messenger RNA. And then you can see that there are these little black spots on the messenger RNA. Those are ribosomes. So these ribosomes are reading this RNA and then producing protein. Okay, so here's an example of measuring the motion of some molecule in E. coli and determining whether it's diffusive or subdiffusive or superdiffusive or what. So this was done by my predecessor here, Ido Golding. Okay, so he was here, he left, and then I got his job. I also came from the same lab that he came from, so I'm like Ido Mark II. All right, so here's the experiment that Ido did. All right, so what he had was a gene that encodes a red fluorescent protein. Okay, so if this gene gets expressed, the cell should light up red. All right, if it's not expressed, the cell will not be red. Now, in addition to this this region of DNA sequence that encodes red fluorescent protein, he included another sequence on the end of this gene that's called the MS2 binding site. Now, these DNA bases do not actually code for anything. What they do is they go through that transcription process I showed you on the previous slide and generates a molecule of mRNA. Okay, now in this messenger RNA molecule, part of it will still code for red fluorescent protein. And then what these things do is rather than coding for something, what they actually do is they fold up and form some specific secondary structure. Okay, so these bases actually, some of the bases in this sequence are actually complementary to each other. And then when you get this piece of single-stranded mRNA, it'll actually fold up and form a series of stem loops. Okay? And in particular, he encoded it to make 96 copies of this stem loop. Now, why did he do this? Well, that's because, so this stem loop actually comes from a virus called MS2. Okay? And this virus, the way the virus is put together is that its genome is encoded as a piece of RNA, just like this. And to protect that piece of RNA, it has a whole bunch of proteins that bind and coat that piece of RNA to form an intact virus particle. Okay, so what that means is that there are proteins that will recognize these sequences and bind to them. And so what Ido did was to take that protein and tag it with GFP. And so then what happens, so he expresses these RNAs, and then he also expresses this MS2 coat protein that's fused to fluorescent GFP. And then what's going to happen is that these proteins are going to bind to these stem loops. And then you will see, what happens is then you'll see RNAs inside the cell as bright green spots. Okay? So here's here's a static picture. You can see that the cells are in fact expressing this RNA and in fact translating into protein because the cells turn red. And you can also see the individual RNA molecules that are produced as these big globs of green. So these globs of green are RNA molecules. So this is a static image. Crap, of course it doesn't work. Luckily, I think I have this exact same image on my lab webpage. I believe we want right here. There it is. Oh, glorious. All right, so what you're looking at here is these bright greens. Can I turn off the lights? Okay. So what you see here, there are actually two E. coli cells. There's one here and there's one here. and then these bright spots correspond to these MS2 coat proteins bound to the RNA molecule. And you can see that they are moving, right? So this guy's moving around, this guy's moving around. And so you can perform exactly the same analysis that we did before on these moving spots. Okay? And here is what Ito found. Okay? So now this is a plot of mean squared displacement versus time. So this is the first kind of plot I showed you. Okay, and what you see is that it's, at least for short times, it's approximately linear, which indicates that it's approximately diffusive. But you can see that at long time scales, it deviates from this linear behavior. Okay, so now, if instead of plotting mean squared displacement versus time, we do this other analysis I showed you, where you plot the logarithm of mean squared displacement versus logarithm of time, this is what you get. So again, all these points are different cells where he measured this mean squared displacement versus time. This line here is where that exponent is equal to 1. This is what you would expect from diffusion, pure diffusion. And you can see that the slope of the diffusion of these RNA molecules is less than 1. That indicates that this is subdiffusive, which tells you, first of all, it tells you that the motion is random. That is, there are no motors that are dragging it around inside the cell. And in particular, it tells you something about how the interior of the cell is organized. In particular, because it's subdiffusive, that means you know that the interior of the cell must be really highly crowded, which we know that it is because there's all kinds of other proteins and junk floating around in there. Yes?
Could you have like a driven molecule like that has a motor on it and thing in like a subdiffusive, like a crowded area and like be able to tell that?
I'm sure it's possible to get any combination of super diffusion and crowded environment to yield essentially any exponent you could imagine. So I would say you would have to do further experiments on this, I think, to determine exactly what's going on. Okay, I think with that I will stop.